Hi, so in this video, I want to talk briefly about protein zip codes. And here we've got sort of a mock proteins. Consider this an amino acid sequence, like an alpha helix, some other amino acids here. Uh, if this is a nuclear protein, you're going to ask yourself, how does the cell know if it's made in a ribosome? How to get this to the nucleus? What we'll have a nuclear localization sequence, so a particular sequence of amino acids that does pretty much nothing else but tell the rest of the cell that this needs to end up in the nucleus. And so we'll call this a zip code, and there will be different zip codes for different proteins that need to end up in different areas. For example, a specific sequence that's involved in shipping something to the nucleus is arginines and lysines all in a row. So you could have a bunch of arginines and lysines in whatever pattern here. It would depend specifically on the gene, whatever didn't disrupt the function of the gene. You have them here on some surface of the protein that's exposed so that you can get an outside protein coming in and binding here. So if you have this sort of random protein here, right, it needs to be able to bind to this area. If it's hidden by some other part of the protein, it's not going to be able to do that. So these protein zip codes allow that to happen.